Hello, everyone, and welcome to today's webinar, Assessment of Quality Attributes of Biotherapeutics Leveraging a Novel Fragmentation Technique. I am Sydney McNeil of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SciEx. To learn more, visit sciex.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Armelle Martelet, Deputy Head of Structural Analysis Lab, Bioanalytics, Sanofi R&D. Armelle, you may now begin your presentation.
Okay, thank you for the nice introduction. I am very happy to have the opportunity to present our work about the assessment of quality attributes of biotherapeutics leveraging a novel fragmentation techniques. So just a few words about who we are. We are from the Sanofi R&D site located in Vitry-sur-Seine in the structural analysis lab, where 11 employees characterize biotherapeutics by mass spectrometry using high and low resolution instruments from various MS vendors. And specifically, we study protein therapeutics, including three main groups: various monoclonal antibodies formats, including multispecific antibodies, and also antibody-drug conjugates, the ADCs. And we also study host cell proteins, HCP, which are process-related impurities that may co-purify with biopharmaceutical drug products. So our main activities, protein therapeutics have unique structural characteristics which contribute to the quality, safety, and efficacy of the product. And these characteristics are known as quality attribute or product quality attribute. And among these, some are critical for effective functioning of the drug, as well as its clinical safety, and they are known as CQA for critical quality attributes. The quality attribute for a given product are defined early in development, and they are progressively refined as additional product knowledge is gained over time with extensive analytical characterization. So our main activities are dedicated to support the product life cycle from early to late stage. And the key message is that we do have to juggle between comprehensive characterization of product quality attribute and their targeted detection with a high throughput capacity, as we may have many samples to analyze. So biotherapeutics are a rapidly growing class with a high diversity and complexity of formats. And in the last years, advancement in antibody engineering enabled formats ranging from nanobodies to multispecific antibodies. And these new generation antibody drugs, which engage multiple therapeutic targets through a single protein, provide advantages for various therapeutic applications with a reduced expense of administering a single biologic therapy instead of complex combination treatment. And such complex protein molecules have a high susceptibility to spontaneous or enzyme-dependent chemical modifications, we called post-translational modification, PTM, and thus an in-depth characterization of the product at the amino acid level is essential to the successful clinical development of biotherapeutic. But this task may be highly complex and sometimes challenging. And among all analytical techniques used for antibody characterization, mass spectrometry plays an important role, such as the bottom-up approaches, which are a powerful tool to provide detailed information on the amino acid sequence and location of PTM. And the rule of mass spectrometry can be reinforced thanks to the impressive advances in instrumentation, such as improvements in ion activation methods that allows even more detailed characterization. And recently, a novel QTOF instrument was released by SciEx, the Zeno TOF 7600 system, with the intuitive and easy-to-use SciEx software for data acquisition. One of the main innovations of the system is the flexibility of multiple fragmentation options, both collision-induced dissociation, CID, and a novel fragmentation technique, electron-activated dissociation, EAD, which brings new capabilities for characterizing PTM. So what is EAD? EAD is a name of a family of dissociation methods that use free electrons at different energies. So how it works? The EAD device is a novel magnetic ion trap cell, which is a branched RF ion trap, and is placed at the front end of the collision cell in the QTOF instrumentation. The device can be operated in a regular ion guide mode or as a trap for ions that are subjected to electron irradiation. There are electrometers which generate free electrons between two magnets in order to focus the electrons. And the EAD device is a reagent-free cell which is pressurized with nitrogen bath gas to facilitate ion trapping. And the electron beam optics were also designed to support ion-electron interaction time or reaction time of 10 milliseconds or longer at the electron energy of 0 for all positive charge states. And such reaction speed will enable LC-MS measurements for multiple applications independent of the precursor ion charge state. And for more detail about this novel technology, please have a look on the great work published by Takashi and coworker. Few words about electron-activated dissociation. Because free electrons are used for the reaction, it is possible to control the kinetic energy of electrons at any value. And it allows for a range of free electron-based fragmentation mechanisms within a single device. As you can see on the right, the device can perform various types of dissociation within a range up to 25 eV, including ECD, using low energy for multiply charge and large peptide and proteins, typically for top or middle down workflows. We can also fine-tune with mid-energies for peptide workflows using hot ECD, and the high-energy EAD fragmentation can produce fragments for singly charged molecules. So the novel instrument enables EAD in an easy and routine way, same as CID. So today, we will look at the fragmentation capabilities of the Zeno TOF system for the assessment of PTM in a biopharma perspective. And because weakly bonded PTM remain intact and the fragments generated by EAD, we can have access to information regarding the location of the sites that are modified, especially for critical PTM. So to that aim, we use the instrument which was recently installed in the lab, and the comprehensive characterization was performed using BIOS from protein matrix with the peptide level analysis mode. So to give you a snapshot of what can be achieved for deep characterization of antibodies, I will present a few case studies about how we can discriminate aspartic acid and isoaspartic acid, either in peptides containing several potential isomerization sites, or after asparagine deamidation generated by thermal stress. Then I will move on case studies about glycosylation localization for both N and O glycosylation. And following that, I'll go through applications on singly charged peptides and finally disulfide bridge peptides. So let's start with applications about the discrimination between aspartic acid and its isomerized version, the ISOASP. because the non-enzymatic isomerization of aspartic acid residues in proteins is considered one of the major PTM contributing to biotherapeutic degradation. And it is important to monitor such PTM during the development of therapeutic proteins. But such detection can be challenging. So just first, I will just start with a few words about this PTM for those who are not familiar. So the isoaspartic acid can result from either deamidation of asparagine or isomerization of aspartic acid, because both residues are subjected to spontaneous degradation reaction depending on the environmental condition and the local conformation, especially if they are in complementarity determining regions, as they are solvent accessible. And such modifications have been reported extensively in therapeutic proteins because they may impact biological function and stability. So according to the generally described mechanism, the reaction proceeds through a nucleophilic attack of the peptide bond on the beta-carbonyl side chain of the residues to form a succinimide intermediate. In asparagine deamidation, this results in the loss of ammonia, while for aspartic acid, it results in the loss of water. And then the succinimide intermediate can be further hydrolyzed into aspartic acid or isoaspartic acid. And isomerization of aspartic acid residues results in the incorporation of an extra CH2 group in the peptide backbone, generating a kink in the conformation with the shortened acidic side chain. And this modification in the protein conformation could alter protein functions. Regarding the detection of these modifications, asparagine deamidation increases the molecular weight by approximately 1 Dalton, so it can be studied using high-resolution mass spectrometry, while isomerization does not change the mass. And one of the most used methods for isoasp analysis is reversed phase liquid chromatography combined to mass spectrometry, if separation can be achieved. And in literature, some studies described slight differences in fragmentation patterns using CID, but the reporter ions are non-specific for isoasp and often require reference peptides to confirm identification. And in contrast, electron-based activation can be useful to study isoasp, and we will see in the next slide how. So here is just a brief reminder of the peptide fragmentation nomenclature. You know that using tandem mass spectrometry, the major sequence ions are formed after peptide backbone cleavages. In the fragmentation nomenclature developed by Ropstoff and collaborators, alphabet letters are used to represent categories of fragment ions based on the type of bond which are cleaved in the peptide backbone. Thus, for dissociation methods such as CID, main cleavages occur at the CN amide bonds to produce B and Y ion series, as highlighted in red on the slide. And the numerical subscript refers to the number of amino acids contained in the fragment ions. And fragment ions that retain the N-terminal side of peptide are called the B ions, while those that retain the C terminus are the Y ions. And in contrast, electron-based activation method induce predominant cleavages between the amino group and the alpha carbon, thus producing C and Z ion series. And because of the incorporation of the extra CH2 group in the peptide backbone of isoasp, a pair of diagnostic ions that are unique of isoasp can be generated using EAD, the C plus 57 and Z minus 57 ions. So saying that, let's see application of a thermally stressed antibody, which was analyzed after trypsin digestion. A peptide belonging to the FC region of the antibody and containing the unstable NG motif was monitored and fragmented by EAD. So extracted ion chromatograms of the non-modified peptide and the deamidated version are displayed on the left, and showing that 2 deamidated peaks were detected. The corresponding EAD spectra are displayed on the right, allowing to see diagnostic ion Z3-57 and C13+57 that were detected in only 1 deamidation peak in the middle panel, highlighted by arrows. Another application concerns the discrimination of several potential isomerization sites within complementarity determining regions, or CDR. Here, a trypsin peptide covering a CDR contains 2 aspartic acid residues at position 4 and position 8. And the LC-MS characterization of the thermally stressed antibody revealed a second peak eluting close to the native version, thus suggesting an isomerization within the peptide. So EAD was performed on both native and modified peptides. So the top spectrum corresponds to the native peptide and the bottom one to the modified version. Both peptides showed good sequence coverage, and if we look more closely, a pair of diagnostic ions, C3+57 and then 17-57 were detected, which is a signature of the isomerization of the aspartic acid residue at position 4. And these ions were not detected in the native peptide, and no diagnostic ions were detected for the residue at position 8. So in that case, EAD allows to confidently identify the aspartic acid residue that is susceptible to isomerization in the CDR. Here, a similar application is shown, and in that case, the trypsin peptide covering the CDR contains 3 potential aspartic acid that could be isomerized at position 6, 8, and 10. Again, the LC-MS characterization of the thermally stressed antibody revealed the second peak, eluting close to the native version, and EAD was performed for both peptides. So the top spectrum corresponds to the native peptide and the bottom up to the modified version. Again, both peptides showed a good sequence coverage. And if we look more closely, the C ions covering each aspartic sites were well detected in both native and modified version, with similar and good intensity for the C5, C7, and C9. And additional fragments were detected for the modified peptide only, with a pair of diagnostic ions C7+57 and Z18-57, which is a signature of the isomerization of the aspartic acid of the aspartic residue at position 8. And no diagnostic ions were detected for the residues at position 6 and 10. So again, EAD demonstrated very good performance to localize ISOASP in a CDR. And we also compared the detection of this pair of diagnostic ions for the thermally stressed antibody and the unstressed antibody. So the extracted ion chromatograms of the targeted peptide are shown on the top for the initial material, and on the bottom for the thermally stressed for 1 month at 40 degrees. Based on this XIC, isomerization was quantified at level close to 4% for the initial material and above 40% for the stressed material. And as you can see on the right panel, the pair of diagnostic ions generated after EAD was detected in the starting material with significantly higher levels than the noise level. Okay, so now I will move on application about glycosylation. So here we are showing at a classical CID-MSMS spectrum from a glycosylated peptide belonging to the FC region of an antibody. As you can see, such data provides mainly glycan composition, but little sequence information and no information about the location of the glycosylation. And now we are showing at the same peptide fragmented by EAD. And in that case, the spectrum provides a lot of fragment ions with preserved glycosylation, as illustrated in the inset. The C ion series from C9 to C12 were successfully detected, allowing confident localization of the modification. Here we studied a peptide from a cynomolgus IgG1 antibody, which has two potential sites of O-glycosylation, a serine at position 6 and a threonine at position 10. The bottom spectrum displays the native version, and the top spectrum displays a modified version with a mass increment of 203 Daltons that could correspond to the attachment of O-linked N-acetylglucosamine. The peptide coverage are shown on the right, revealing excellent coverage for the modified version, with a modification located on the threonine. So let's have a closer look into the spectrum to confirm this localization. So first, the fragment ions C3 to C6 were nicely detected without any modification, with the C6 ion covering the serine. There, the unmodified C10 ion, which cover the threonine residue, was well detected in the native peptide and was detected with the intact glycan in the modified version, as well as additional C ions such as the C21, C25, or C30. And finally, if we look at the ions retaining the C terminus, especially the Z27, it was well detected in the modified peptide, only, with the intact glycan. So it removed any doubt on the localization of the O-linked glycan on the threonine. As mentioned earlier during the presentation, the EAD device can produce fragments for singly charged molecules. And one interesting application concerns the sequencing of the N-terminal or C-terminal peptides of antibodies. Here is an example of a C-terminal peptide, a singly charged peptide, of course. EAD provided good sequence coverage, mainly obtained with the B ion series. Another example is related to a 5 amino acid peptide. In that case, the peptide was identified with good sequence coverage, but at two different retention times in its native version on the bottom and a modified version with an increase of 80 Dalton, suggesting a potential phosphorylation or sulfation. So the top spectrum corresponds to the modified version, and I will show you a couple of slides with more detailed data to provide a confident characterization of this modification of 80 Dalton. So first, peptide mapping data were searched with both phosphorylation and sulfation, which has very close masses. So experimental MZ ratios were measured on a series of 4 injections repeated over 3 days, and results are summarized in the table. Results enable to match phosphorylation with 30 ppm or a sulfation with less than 1 ppm error. So it's likely that we have a sulfation, and we are quite confident about that because the instrument is equipped with a heated TOF, giving a high mass accuracy, and we are also able to perform automatic external calibration before each injection. But to confirm the identity of the PTM, a desulfation study was performed using a specific enzyme able to remove sulfation, the sulfatase type 8. So on the top, you can see the extracted ion chromatogram of the sulfated peptide, the blue trace, just here, eluting around 26 minutes with the corresponding mass spectra on the bottom. And the unsulfated peptide, which is the pink trace, eluting around 21 minutes and the corresponding mass spectrum. If we look first at the negative sample, which is the left panel, and which corresponded to the antibody mixed with buffer instead of the enzyme. So we measured a low level of unsulfated peptide, around 3%. And now if we look at the positive sample, the right panel, which correspond to the antibody mixed with the enzyme, we observed up to 80% of unsulfated peptide, indicating a successful desulfation event. Thus, the identity of the PTM was confirmed for this peptide. We are speaking about a sulfation. However, there is still an unsolved question about the localization because sulfation occurs at tyrosine residues and the targeted peptide contains 2 potential tyrosine sites that could be sulfated. So that's why EID experiments were performed to provide information about the localization combined to the use of a synthetic peptide. So the targeted peptide contain 2 potential sulfation sites at position 2 and at position 5. First, we are looking at the antibody digest with the EID spectrum on the left and a zoom on the Y2 fragment that contains tyrosine at position 5, modified with the sulfation. We performed similar experiments with synthetic peptides. The middle panel with a modified peptide with a sulfation at position 5, and the bottom, an unmodified version of the peptide. And results clearly demonstrated the absence of the characteristic Y2 fragments in the unmodified synthetic peptide, whereas it was detected in the sulfated synthetic peptide. So results confirm that the sulfation is located at position 5 in the peptide from the antibody digest. Okay, and just for the last case study, I would like to give you a quick snapshot about disulfide bridge peptides. So EID experiments using disulfide bridge peptides allow to provide high sequence coverage, but also confirm the identity of both linked peptides thanks to the cleavage of the disulfide bridge. Here are 2 examples where fragment ions corresponding to each individual peptide were well detected and highlighted with arrows. And there, just 2 other examples illustrating successful fragmentation and high sequence coverage, even for large peptides. So to conclude, I have shown various case studies illustrating the capacity of our platform to characterize next-generation biotherapeutics, which combines easy and tunable data acquisition on the Zeno TOF system and the software BIOS from Protein Metrics to process the data, thus allowing a comprehensive and confident characterization. And during the presentation, I also gave you a snapshot of what can be achieved using the alternative fragmentation method EID for the confident localization of glycosylation, ISOASP within CDR, the ability to fragment singly charged peptide while preserving labeled PTM such as sulfation, and finally for disulfide bridge peptides. So I finish with the thank you slide. I would like to thank the Sanofi member from the MS Lab, especially Valérie Garrigue and Séverine Beauvisage, who were highly involved in contributing to the great data shared today. And I also would like to thank everyone from SciEx team who has supported us during the last months, including the service engineer and MS experts, and also Jason, Zoe, Sibyl, Todd, and Elliot, and also a special thank you to Kirsten. And thank you all who joined the presentation today for your interest and your time, and I would be happy to answer some questions.
Thank you, Armelle, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, EAD sounds very interesting, but how reproducible is it?
Yeah, that is an interesting point. Actually, it is still under evaluation using an internal system suitability test. Actually, we are monitoring a couple of peptides bearing critical PTM, and we are looking at their EAD fragmentation pattern, meaning we pick up some signature fragments and report their mass and intensity. And what I can say today is that preliminary results are very promising. Over approximately a 4-month period, we found very consistent results. So just let's wait a few months to have more data and properly answer that question.
Perfect, thank you. Our next question is, which EAD energy conditions did you use in the presented case studies?
Up to now, for peptide mapping workflow, we used in first intention energy conditions close to hot ECD with approximately 7 EV. And we observed that such conditions were applicable for most peptides from antibody digest. So it prevents the need for special tuning and enables a quick way of getting a lot of information. However, if there are very challenging cases, such as large disulfide with peptides, the electron kinetic energy can be optimized and decreased. For instance, we decrease the energy as low as 1 EV for some large peptides.
Wonderful, thank you. Our next question is, what is the difference of EAD compared to ECD or ETD?
Well, the main differences are related to kinetic energy and time of irradiation as well. The reaction time is very short, in the low millisecond range, whereas ETD takes way longer. And that impacts its usage for data-dependent acquisition. Second, the difference also occur also related to the efficiency. It's highly efficient as it is not a flow-through device, but a magnetic trap. And finally, it's about also the tunability. EAD can be tuned from ECD to hot ECD to EIEIO. Thus, EAD is the name of a family of dissociation methods that use electron at different energy. And another aspect is also related to the instrument handling. No extra reagent is needed for EAD fragmentation. The cell is only pressurized with nitrogen bath. And in addition, the Zeno TOF is also an easy-to-use system, which is managed by the intuitive ISOAS. And for those who are already familiar with the X500B instrumentation, that is exactly in the same way, with additional things that are EAD specific, of course.
Thank you. Our next question is, how much on-column load do you need to get meaningful intensities, especially compared with CID?
Up to now, we worked with the same sample load that is used for our routine CID injection, with approximately 5 micrograms of protein digest. And we got satisfactory results. What I can say is that using EAD, the precursor ion is not completely consumed, so it limits internal fragmentation and facilitates spectral annotation. And in the case you need a bit more precursor, you can also use the Zeno trap on demand, which enhance the MSMS intensity whenever the precursor intensity is below a certain threshold. And even if the fragment ion intensity are quite low, the noise level observed on our spectra is generally very low, much lower than observed for CID. And a specific tuning module is available inside ISOAS and dedicated to the background noise reduction in a few minutes before your data acquisition.
Perfect, thank you. Our next question is, do you think EAD could be a replacement method for CID for peptide mapping? Why or why not?
Yes, that is an interesting question, and we could probably debate about that for some time, but I will try to provide some elements to answer that. To my opinion, I think we should keep in mind that both fragmentation methods are complementary. The different fragmentation mechanisms that are involved allow to provide a detailed coverage of peptide sequence, thanks to the peptide backbone cleavages with BY ion on one side and CZ ion on the other side. And the combination of spectral information from both CID and EAD could be useful for confident characterization of the variable domain of antibody, especially the CDR zones, and even for de novo application. But speaking of routine characterization, one of the main advantages of CID is to be highly efficient, robust activation, and that's why it's a gold standard for many routine proteomics applications. And disadvantage in that such slow heating activation method favors fragmentation that result in the predominant cleavage of the weakest bonds. So it is often associated with loss of informative data because of the frequent neutral loss of PTM, water, or ammonia. And that limits the usefulness of CID for accurate localization of PTM. And on the contrary, with EAD, you can have a direct answer for accurate localization of labeled PTM. And that is a great advantage over CID. But I think we need to provide a few additional elements to properly answer that question. I mean, we need to demonstrate, for instance, that sequence coverage performance are at least equivalent to CID for our biotherapeutic application, and that on a large data set. And that point is still under evaluation, and preliminary results are very satisfactory. So it's a bit early to answer that question. We are collecting data from various antibody digests and looking at the sequence coverage performance, also including an evaluation of the sequence dependence of the cleavage. As you probably know, upon collision activation, sequence-specific backbone cleavage can limit information from peptide containing proline or aspartic acid. So we need to pay attention to such consideration to evaluate if preferential cleavages are not as significantly enhanced as observed with CID due to amino acid composition. And besides, the evaluation should also be performed for complex samples, such host cell protein extract, so that a complete picture could be provided. Well, if I try to summarize, I would say, yes, EAD could replace CID as method used in a first intention for our future projects, and then be completed with CID for specific application, provided that the additional elements I mentioned before are properly evaluated and confirmed on a large data set.
Perfect, thank you. It looks like we have one final question. What added value could this instrumentation bring to your activities?
Considering that EAD allows to accurately pinpoint critical PTM on amino acid residues, I see a lot of added values for our activities. Speaking of in-depth characterization of critical quality attributes, a more detailed understanding of the complementarity determining regions, provide confident data for health authorities, also help refining protein engineering for second generation product with identification of amino acids that could be mutate. So definitely having the Zeno in the lab is an added value. considering that the other instrumentation we currently have in our lab, only the Zeno allowed to localize isoasp or O-glycosylation with such confidence.
Perfect. Thank you. Thank you again, Armelle, for your time today and your important research. We would also like to thank LabRoots and our sponsor for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.